Alright, so in today's presentation I'll be talking about DNA and protein synthesis. So, as you can see on the title page here, I have DNA and protein synthesis, and I have the central dogma, and that is DNA to RNA to protein. And that is the central dogma of biology, which states that DNA is transformed or transcribed to RNA, and then RNA is translated to protein. So that simplified, it's DNA to RNA to protein, and that is what we will be talking about today. So before I begin, here are the topics that will be covered in this presentation, and you can go ahead and click on any one of those that you'd like to study. You might not need to study every single one of them, so I've provided links so you can skip to that portion of the presentation. But the first topic we will cover is DNA, and just giving a brief overview of DNA. So before I truly begin, I'll mention now that I'm going to be using an analogy between DNA and some of the other elements I'll be talking about and a book or languages. So to begin, DNA is like a language that only uses 4 letters, and those 4 letters are A, T, G, and C. Those letters are nucleotide bases, and they bond together in very specific ways. For example, A only ever bonds to T, and G only ever bonds to C, and they're bound with hydrogen bonds. And that those arrangements of nucleotides, the sequence that they're in, determines what the gene will express, and at the end of the day, at the end of the whole process, going back to the central dogma, the sequence of nucleotides will determine the shape of the protein and what the protein does, but we'll see that a little later on. Structurally, DNA is normally found in a double helix with sugar phosphate groups as the backbone, and what that means is that when in a double helix form, the nucleotide bases, the letters, will be in the middle, and the sugar phosphate groups will be in the back, acting as the backbone. And you can see that here. In the first diagram, the anti-parallel DNA strands diagram, you can see clearly the sugar phosphate groups represented by the blue pentagons, and you can see the C's bonding to G's and the A's bonding to T's. In the middle, those red dotted lines represent the hydrogen bonds, and you can see that between the C's and the G's there are 3 hydrogen bonds, and between the A's and T's there are only 2. And that is why a T will never bond to a C, and an A will never bond to a G or a C. In the second diagram, the DNA ladder, it's a little more clearly shown how the A's and T's and G's and C's fit together, as well as the sugar phosphate backbones. It's a little simplified. And the last diagram shows the double helix structure. It's kind of wound up. So the structure of DNA wasn't always known, and it's Watson and Crick who take the credit for discovering the structure of DNA. However, Rosalind Franklin, the woman pictured on the right, was actually the one who found the final piece of the puzzle and completed the work that Watson and Crick had done in discovering the true structure of DNA. She took that X-ray image of DNA, and what that revealed was that it was the sugar and phosphate groups that were on the back and the amino acids in the middle, and not the other way around, as Watson and Crick had suspected. The next topic is DNA transcription. We're moving right along with our central dogma. This is when DNA goes to RNA, and it is the first step to writing a book using DNA. So transcription essentially changes DNA into mRNA, which is a single-stranded polynucleotide. DNA is a double-stranded polynucleotide, so mRNA is quite a bit simpler. mRNA is complementary to DNA, and in mRNA, every T is replaced with a U. So every T nucleotide is replaced with a U. And that's important. That's different than DNA, where T's are still T's. So to do this, to transform or transcribe DNA into mRNA, an enzyme, which is made of protein, the enzyme RNA polymerase travels along the length of the DNA and adds complementary bases. So if the strand of DNA progresses as ATTGCCGA, those are the nucleotide bases, the mRNA strand would be complementary to that. So the mRNA strand would be UAACGGCU, because T's are now replaced with U's. After this complementary strand is made and the RNA polymerase detaches from the DNA strand, after it stops transcribing, the mRNA strand will exit through a nuclear pore and head on its way to a ribosome. So here is a diagram. You can see RNA polymerase is represented by the gray circle. It's moving towards the 5' end of the DNA strand, and the mRNA is being transcribed beginning at 5' and going to the 3' end. And you can see here the adding of the opposite bases. So the DNA strand is CATG and the mRNA strand is GUAC. So that is the opposite, the complementary strands, the complementary nucleotides being added. So each portion of the DNA that's transcribed is essentially like a sentence, and this adds very important information to the book. RNA polymerase, the enzyme mentioned earlier, it begins at a promoter region, and that's a specific sequence of DNA, specific nucleotide sequence, that tells the RNA polymerase that is where the gene begins. That's where transcription should begin. And similarly, at a terminator region, that's where RNA polymerase is told to stop transcribing, that the gene is over. You can think of a promoter region as a capital letter and a terminator region as a period, because a capital letter always begins a sentence or a gene, and a period always ends a sentence or a gene. The next topic is ribosomes and mRNA translation. So this is where we go from the RNA to protein. And this is a very important step, because without it, the whole process would be for nothing. Ribosomes are proteins. They have two subunits, and they translate the mRNA sentences that were just made by RNA polymerase, and they translate them into a language that our cells can read and understand. And that language is different than the DNA language and the mRNA language. This language uses amino acids. So essentially, ribosomes are translators that translate the mRNA into sequences of amino acids. And these amino acids are more complex than just DNA or just mRNA, and long sequences of them can be made to achieve different forms of peptides and proteins. Peptides and proteins are the result of this whole process of DNA to RNA to proteins. That's the end of the dogma right there, peptides and proteins. And these amino acids, these components that are going to build the proteins, are brought to the ribosome, to the translator, by transfer RNA, tRNA, which carries them to the site of translation. And when translation is done, the instructional book for the cell is made. It's finished. That is the end product. So here, this picture is a chart showing all of the different amino acids that can be made. So to read this chart, you look at the first letter in the codon, and the codon is a sequence of 3 letters, 3 nucleotides on the mRNA. Yeah, so you start at the first letter of the codon. In this example, we'll look at A. Then you go to the second codon, and so we'll look at G for this example. And to finish, you go to the third codon, and in this case, if I picked G or A, it would be arginine, and if I picked C or U, it would be serine. So essentially, a codon is a section of 3 nucleotides, and that's how mRNA is read. It's not read letter by letter, but in chunks of 3, and each codon, or chunk of 3 letters, 3 nucleotides, each codon corresponds to one amino acid. So if one of those letters are changed in the codon, it can result in a mutation, and we'll talk about those later. But if a mutation occurs, if the sequence of the sequence of nucleotides changes, the resulting amino acid can change as well, and that will disturb that, or it could disturb or disrupt the resulting protein or peptide by impacting how it folds or how it bends. And since the shape of a protein determines its function, it can change the entire function of the protein as well. So here is a diagram explaining exactly how tRNA, transfer RNA, brings in the different amino acids into the ribosome. So as you can see, this teal zipper looking thing, that's the mRNA. That's what we got out of the process of transcription. And it's going through the ribosome, and the ribosome is reading it and collecting or drawing in the tRNA that corresponds to each codon. You can see that each yellow tRNA rectangle has three bumps at the end, and that is showing us how mRNA is read in chunks of 3. It's read in codons. ACC is a codon, UUU is a codon, CUA is a codon. And on each of those tRNA, represented by the pink circles, those are the amino acids. And you can see that as more tRNA segments are brought in, so are more amino acids, and it builds the chain, it builds that peptide, it builds that protein. So now let's talk about the difference between peptides and proteins and what they do and why they're important. So after translation, after mRNA has been read, after the amino acids have been built, the book is essentially finished for the cell to read. That means the protein or the peptide has been made, it's been formed, the cell can use it to carry out important processes, and we have achieved our goal. Hopefully, if no mutations have occurred, then the protein and the peptides are the right shape, they've got the right amino acids, and they're able to do what they are meant to do. So the book that's finished, the cell can use that, the cell can use the protein and the peptide to do what it needs to do. Proteins and peptides are very similar. Proteins are generally longer, more complex than peptides, and they have a more defined structure, which can either be secondary, tertiary, and quaternary structures. And as you go up and up, the structure gets more complex. So peptides can't do that. They don't have that defined structure. One special thing that proteins can do is form enzymes, and enzymes are super important. They carry out a myriad of different reactions throughout the cells and throughout the body. So they act as catalysts for chemical reactions. They lower the activation temperature and the energy needed to carry out a reaction. But it's important to distinguish between proteins and enzymes, because not all proteins are enzymes, but all enzymes are proteins. And that's important. So here is a little bit about how enzymes work. So you can see in the green, that's a substrate, that's the beginning of the reaction. That's what you have in the beginning, and that binds with an enzyme that has a similar shape. It has to have a complementary shape to be able to bind. So if the shape of the substrate or the shape of the enzyme, the active site on the enzyme changes, then that substrate will no longer be able to bind, and the enzyme will be prevented from carrying out what it is intended to carry out. And that is why a mutation can be so deadly, or so damaging, is because a mutation can change the sequence of amino acids, and it will change the shape of the active site on the enzyme or protein. But if everything is working well, the enzyme and the substrate will bind together, and the product or products will be formed. In this case, a green oval and triangle are binding with an enzyme that has a complementary active site, and they're being split apart. And so that is the function of the enzyme. And as mentioned earlier, another thing enzymes do is lower the energy of the activation energy, and they catalyze. Without enzymes, reactions would have to occur at a higher temperature, and such high temperatures aren't easily found in the human body or in the bodies of animals and other organisms, because such temperatures can denature proteins. They can break apart proteins and break apart the enzymes themselves. So it would be very dangerous to have such a high temperature for all of these reactions, and that's where enzymes come in. And you can see on this graph here, the amount of free energy used is much less in a catalyzed reaction than in an uncatalyzed reaction, and that's a very important thing for organisms to have. So I've mentioned mutations a few times, but let's dive right in. So if any letter in any sentence becomes changed, the resulting instructional book, the resulting protein, could have a different meaning, it could perform a different function, or it may be defective entirely. And there are two basic types of mutation. There are point mutations and insertions and deletions. Point mutations affect a single nucleotide, and generally it's a replacement. And insertions and deletions are pretty self-explanatory. Insertions are an insertion of a nucleotide or multiple nucleotides, and deletions involve the deletion of one or more nucleotides. And such insertions and deletions can change the reading frame for the tRNA. And this means that the codons will be changed and different amino acids will be brought in. There's a diagram of that coming up that will explain it much easier. But some of these mutations, even though they change the amino acid or they change the codon, they are silent. They don't impact the resulting protein or peptide. So you or I could be walking around with mutated DNA or mutated RNA, and we could be perfectly fine and never know about it because it's a silent mutation. Generally, mutations occur in the DNA after replication or as a result of multiple different things, but in the end, they can result in a wide variety of effects. Here are the different types of mutations. On the left, we have point mutations and insertions and deletions. Those are pretty straightforward, as I explained earlier, but frame shifts are a little harder to understand. So the top of this in blue those are representing the amino acids which are meant to be a result of these codons. However, if you insert a C between the T and the G in the second codon, that shifts the frame so that we have an extra, if you will, nucleotide on the end, and the codons that we have are read differently than they were before. This insertion has changed TRP to SER, and all of the rest of the amino acids are different than what they would have otherwise been if there were no mutation. This is perhaps the most damaging mutation because it's so widespread and it affects the whole peptide and the whole protein. One complicated element of this subject, this DNA and protein synthesis, is the concept of introns and exons. Not all of the letters from the DNA are used. Not every part of that sequence is used. Some of that sequence and some of those letters are silent and they're cut out entirely. Introns are not transcribed and they are not translated. Exons are. Exons affect the resulting peptide and protein. Introns do not. Occasionally, exons may be eliminated from the mRNA sequence, and that is called alternate splicing. And alternate splicing allows a single gene to code for multiple proteins. That's important because we can cut down on the amount of genetic information that's available to be mutated or that has the potential to become mutated. So this flow chart shows an example of introns in the blue area being cut out and the exons being spliced together, and shows how the introns have no impact on the resulting mRNA sequence, and it is the exons, the coding sections of the DNA, that make up that mRNA. Alternate splicing takes those exons and it combines them in different ways. In protein A, you have exons 1, 2, and 3. And in protein B, you have exons 1, 2, and 4. And that results in a completely different protein with a completely different shape than before. And that will result in a protein that bends and folds differently than the other protein, than protein A, and a different function, therefore, as well. So, in summary, we have proteins and peptides, and those begin as DNA. DNA is transcribed by RNA polymerase into mRNA, into messenger RNA rather. This mRNA travels to a ribosome, which translates it into peptides and proteins by reading codons on the mRNA sequence. The tRNA brings amino acids into the ribosome and builds up the chain of amino acids, and this amino acid chain bends and folds and creates bonds, and these bonds determine the shape of the protein or peptide. It is the shape which determines the function of the protein or the peptide. If a mutation occurs, it has a potential to be silent, but it also has the potential to change the structure and therefore the way that the resulting protein or peptide bends and folds, and the shape and how it interacts with its environment and its function. So here is an overview of the entire process, starting with DNA, going to transcription into mRNA, which we talked about, and tRNA, which we talked about, and those both go to a ribosome to be translated into amino acids and finally proteins. So I hope this video helped. If you need to review, I've linked the presentation I used for this video in the description. So good luck and study hard.